Precision medicine is definitely the future of cancer research. It's the future of cancer treatment. It's really fundamental. We're really trying to understand what's really driving the cancer in individuals. Genomics and genetics is really now a key component of how we care for cancer patients. It's really important that patients know what's unique about their disease, what are the mutations that are driving their cancers, because that's in many cases allows us to tailor the treatment to their genetic makeup. We think that the work that we do here at the Englander Institute for Precision Medicine is very unique in many ways. One, we are using better technology to sequence people's genomes. We're sequencing the entire genome of individuals with cancer, really looking very hard for mutations. Another thing that's very special here is we try to grow what we call tumoroids. This is the idea that you can take cells from the patient, living cells from the patient's cancer, and you can grow these cells in a dish and make a lot of mini tumors that are essentially copies of a patient's tumors. And using robotics, we can screen these tumor cells for drugs that can kill those cells in a very effective way, and in a way that's unique to each individual patient. So that's a tool that we're hoping to at some point bring to patients. It's still experimental research level, but we're working very hard to make this tool available as a way to treat patients in the future. The other thing that we're doing here is to go beyond just DNA sequencing. We have a program where we can take cells from a patient, grow these cells in a dish, make a lot of mini tumors that are essentially copies of a patient's tumors, and we can treat each of these mini tumors using a different drug. And essentially what we do is a mini clinical trial for individual patients that allows us to test all kinds of different drugs without having to treat the patients. And once we find a drug that works in these mini tumors outside of a patient's body, then we can treat the patients accordingly. As we've sequenced now lots of genomes, we realize that very often there are mutations that we find in cancer genomes, in people's genomes, that we did not expect to find. For example, not too long ago, we found a patient with bladder cancer. When we sequenced the genome, we actually realized that this patient had a mutation that you normally don't find in bladder cancer, but you find in breast cancer. And in breast cancer, it turns out that there's actually a drug that can target the product of that mutation. So what we did for this patient was to treat the bladder cancer patients with the breast cancer drug. And because this mutation was very important in the bladder cancer patient, the drug actually was very effective in that patient. We're moving away from defining cancer as where it's located in the body towards really understanding what's driving the cancer, what's causing the cancer in the first place. And that's very helpful in terms of understanding and predicting what's going to be effective in individual patients in terms of drugs and treatments.